Okay, let's unpack this. Right now, as you're listening, your body is engaged in a silent, constant battle.
It really is.
Microscopic invaders, bacteria, viruses, toxins, they're everywhere, trying to breach defenses, trying to cause chaos. Yet most of the time, you're utterly unaware of this war.
Mm-hmm.
Why? Because you possess an incredible, sophisticated capability, immunity.
That's the core idea, isn't it? Immunity is the body's remarkable resistance system, always sort of on patrol, identifying potential threats and neutralizing them before they can really take hold.
Yeah.
It's the reason we navigate a microbe-filled world without constantly being overwhelmed by illness.
Absolutely. And today we're taking a deep dive into your sources, the material you've shared to truly understand the mechanics of this system.
Right.
We're going to pull out the key insights, the fascinating details, and aim to understand how this intricate network provides such powerful, specific protection, almost like looking at it through the eyes of someone who needs to really master this field.
Exactly. Our mission is to go deep into this material, focusing on every concept, every mechanism described. We want to understand not just the general defenses, but the astonishingly specific responses, how they're initiated, how they amplify, and yes, what happens when, in rarer cases, this incredibly powerful system misfires.
So let's start with the fundamental definition the source provides. Immunity is defined simply as the body's ability to resist almost all types of organisms or toxins that tend to damage tissues and organs. It's resistance to those damage-causing agents.
And what the source immediately highlights is that this resistance isn't a single uniform process. It's broadly categorized into two main types,
Which are acquired immunity and innate immunity. And they're fundamentally different, right, in how they operate and how quickly they respond.
Totally different. Let's start with acquired immunity. This is the highly specific, incredibly potent defense system that you develop over time. It kicks in after your body has been initially exposed to a specific invader, maybe a particular strain of flu virus, a specific type of bacterium, a unique toxin, or even like foreign tissue.
So it's not something you're born with fully formed against every possible threat. It's a learned response.
Precisely. And because it's a learned, tailored response, it's not immediate. The source points out it often takes weeks or even months for this specialized acquired immunity to fully develop its peak power against a new threat.
Weeks or months.
Yeah, think of it as building a custom weapon specifically designed to target one particular enemy blueprint. It takes time.
Okay, contrast that with innate immunity.
Yeah.
This is your body's built-in, ready-to-go defense system. These are general processes. They are not directed at specific organisms or toxins.
Right. Innate immunity provides immediate, broad, non-specific protection. It's like the general security measures of a building, locked doors, alarm systems, general cleanup crew.
Right.
They deal with many types of potential issues without needing specific training for each one.
So you're born with innate immunity, providing that initial shield, while acquired immunity is something you build over your lifetime through exposure, offering powerful targeted resistance.
And both are absolutely indispensable for survival. You need both.
Definitely.
And the source gives us a wealth of detail about the components of this innate immunity, this first line of defense that's always operational. It's not just one barrier, it's a multi-layered system.
Let's walk through what's listed. First, there's phagocytosis carried out by white blood cells and cells of the tissue macrophage system.
Yeah, the engulfers.
The source mentions this is linked to other chapters, but focusing just on what it does. These are specialized cells that can engulf and digest foreign particles, bacteria, cellular debris.
They are the body's cellular cleanup and initial response force, like little Pac-Men.
Yeah. Then you have chemical defenses, like the destruction of swallowed organisms by the highly acidic secretions in the stomach.
That stomach acid is powerful.
And the various digestive enzymes present in the gastrointestinal tract. This is a critical barrier for anything entering through the mouth.
Absolutely critical. Keeps a lot of stuff out.
The skin itself is a massive component of innate immunity. Its physical structure, the layers of keratinized cells,
Just a physical wall, basically.
provides a mechanical barrier that is remarkably resistant to invasion by most organisms. Healthy, intact skin is a formidable wall.
It really is. Don't underestimate good skin integrity.
And beyond these obvious barriers and cellular cleaners, the blood contains a variety of chemicals and cells specifically equipped to destroy foreign invaders or toxins.
Right, the internal defenses. The source lists some key examples here.
Like lysozyme. This is described as a mucolytic polysaccharide. Its function is quite specific. It attacks the cell walls of many bacteria, causing them to undergo dissolution.
So it breaks them apart.
Essentially breaking them apart. It's a chemical agent targeting bacterial structure, found in tears and saliva too, right?
Yeah, it's in secretions. Then there are basic polypeptides. These chemicals in the blood can react with and inactivate specific types of gram-positive bacteria.
So a bit more selective within the innate system.
A little bit. It's a slightly more selective chemical weapon within the broad innate arsenal.
And then there's the mention of the complement complex. The source notes, this is a system involving around 20 proteins.
Yeah, we'll definitely talk more about complement later. It's huge.
It's always present in the blood and tissue fluids waiting to be activated. While we'll deep dive into its powerful mechanisms later, it's listed under innate immunity because the system itself is constitutively present and ready.
Exactly.
Even though its full power is often unleashed by acquired immunity. It can also be activated directly by pathogens sometimes.
Right, it bridges the two systems in a way. And finally, we have natural killer, or NK, lymphocytes.
NK cells.
These are a type of lymphocyte that belongs to the innate system because they don't require prior sensitization to a specific antigen. They can recognize and directly destroy certain foreign cells, tumor cells, and cells infected with viruses.
How do they recognize them?
By recognizing general patterns of abnormality or non-self, rather than a specific antigen shape like the acquired system does.
Got it. So innate immunity is a combination of physical barriers like skin and stomach acid, chemical agents like lysozyme and polypeptides, cellular engulfment by phagocytes, the ready-to-go complement system, and these patrolling NK cells.
It's quite an array of immediate defenses.
It really highlights that the body isn't waiting for a specific threat to build some defense. It has multiple general strategies already in place.
Exactly. And the source gives us some really clear examples of how effective this innate resistance can be, even at the species level.
Right, like how humans are innately resistant to certain animal diseases, paralytic viral infections that affect animals but not us, hog cholera, cattle plague, distemper in dogs.
We just don't get them. Even with exposure, our biology isn't susceptible.
And conversely, many animals are resistant or effectively immune to diseases that are devastating to humans, like poliomyelitis, mumps, human cholera, measles, syphilis.
Yeah, it's not about past exposure in an individual animal. It's about a fundamental difference in their biology that provides innate resistance to these specific human pathogens.
It's a powerful demonstration that not all organisms can infect all hosts. The kind of species barrier.
That's a great way to put it. So while innate immunity provides the immediate broad strokes defense, the real specific heavy-hitting power comes from acquired or adaptive immunity.
Right. As the source says, this develops specifically against individual invading agents.
Yes. Targeting lethal bacteria, viruses, toxins, or even foreign tissues from another individual or species. And the key characteristic reiterated here is that it is extremely powerful and specific.
How does it achieve this? The source explains it's caused by a special immune system that forms either antibodies or activated lymphocytes, or both.
Right, and these then attack and destroy only the specific invader that triggered their formation. Laser focus.
And the degree of protection this system can offer is astonishing. The source gives the example of protection against toxins like botulinum or tetanus toxin.
Yeah, this is amazing. With acquired immunity, a person can be protected against doses up to 100,000 times the amount that would be lethal without that immunity.
100,000 times. That number is just mind-blowing. It really underscores the immense destructive potential these toxins have.
And the equally immense protective power the body can mobilize specifically against them. Its incredible neutralization capacity.
And it's precisely this extreme specific protection that is the basis for immunization.
Exactly. As the source notes, immunization is a process used to intentionally induce this acquired immunity to protect people against devastating diseases and toxins. We'll definitely explore the mechanics of that later.
Okay, so this acquired immunity is powerful and specific. The source then breaks it down into two closely allied types.
These are humoral immunity and cell-mediated immunity, two main branches.
Humoral immunity is also known as B-cell immunity. This is where the body develops circulating antibodies.
Right. These special globulin molecules found in the blood plasma, they are produced by B lymphocytes.
And the antibodies themselves are the active agents that attack the invading organism or toxin.
So with humoral immunity, the B cells are the factories, producing soluble weapons, the antibodies that are then released into the bloodstream and tissue fluids to find and neutralize the enemy.
Exactly. The second type is cell-mediated immunity, or T-cell immunity. This is achieved through the formation of large numbers of activated T lymphocytes.
Okay.
These lymphocytes are specifically crafted in the lymphoid tissues to destroy the foreign agent.
So in cell-mediated immunity, the activated lymphocytes are the attackers themselves. They are cells produced by T lymphocytes that physically seek out and directly interact with the target to destroy it.
They don't release soluble weapons in the same way. They do the work directly.
Correct. It's a direct cell-on-cell attack.
So we have B cells leading the humoral antibody-based attack and T cells leading the cell-mediated direct cellular contact attack.
And the source highlights that both types of antibodies and activated lymphocytes are formed in the lymphoid tissues of the body.
Like lymph node spleen.
Thymus, bone marrow, yeah. This is where the acquired immune response is largely orchestrated, the command centers, if you will.
Now, to develop this specific acquired immunity, the system needs a trigger. It only responds after an invasion. That trigger comes from antigens.
Right, the antigens are the flags that say, I'm foreign.
What exactly is an antigen in this context? The source defines them as specific chemical compounds that are part of the invading organism or toxin.
These compounds are recognized by the body's immune system as being different or foreign, not part of the body's own makeup. They initiate the acquired immunity response.
What makes a substance antigenic? The source gives us the key properties. Antigens are usually large molecules.
Typically, proteins are large polysaccharides. Size matters here.
And they generally have a high molecular weight, usually 8,000 or greater. But size isn't the only factor, is it?
No, crucially, they must also have specific molecular groups on their surface that are regularly recurring. These are called epitopes or antigenic determinants.
Epitopes, okay.
The source explains that these epitopes are the specific sites on the antigen that the immune system actually recognizes and reacts to, like the specific features on a key that fit the lock.
So it's a combination of being a large protein or polysaccharide with specific repeating molecular shapes, epitopes on the surface, that allows the immune system to identify something as foreign and launch a specific response.
Exactly. This is why substances like lipids, for example, aren't generally antigenic on their own. They lack that kind of complex recurring surface structure.
This specific recognition of epitopes is fundamental to how acquired immunity achieves its precision.
Absolutely key.
Okay, so antigens are the triggers. Now, who are the cellular maestros conducting this acquired immune response? The source is very clear. Lymphocytes are the essential architects of acquired immunity.
Absolutely essential. Without lymphocytes, whether due to a genetic defect where they fail to develop, or destruction by agents like radiation or certain chemicals.
Like in some immune deficiencies.
Right. Acquired immunity simply cannot happen. The source paints a stark picture. Such individuals would rapidly succumb to overwhelming bacterial infections.
It underscores just how vital these cells are for human survival. We literally couldn't live without them.
Pretty much. So where are these lymphocytes strategically located in the body? The source tells us they are found extensively in the lymph nodes.
Makes sense. Filtering the lymph fluid.
But also in other critical lymphoid tissues like the spleen, the submucosal areas of the gastrointestinal tract, like Peyer's patches, the thymus, and the bone marrow.
The distribution is key, isn't it?
Yeah.
The source highlights that this strategic placement allows them to intercept invading organisms and toxins before they can spread widely and cause systemic damage.
Exactly. Lymphoid tissue in the gut wall, for example, is perfectly positioned to screen antigens absorbed from the digestive tract.
And tonsils and adenoids in the throat catch invaders from the upper respiratory tract.
Lymph nodes filter lymph from peripheral tissues, and the spleen and lymphoid tissue in the bone marrow monitor the circulating blood. It's a networked defense system covering entry points and major pathways. Very clever design.
Within these lymphoid tissues, even though they might look similar, the lymphocytes are divided into two distinct populations: the T lymphocytes and the B lymphocytes.
T lymphocytes are the ones responsible for cell-mediated immunity, forming those activated killer and helper cells we mentioned.
And B lymphocytes are responsible for humoral immunity, the ones that will eventually produce antibodies.
Now, both types originate from the same source: multipotent hematopoietic stem cells in the bone marrow,
The source of all blood cells.
which give rise to common lymphoid progenitor cells. But these progenitor cells aren't ready for prime time. They need further processing or differentiation in specific organs before they are functionally mature. They need training.
This preprocessing step is absolutely critical. The original stem cells or progenitor cells can't directly form activated T cells or antibodies. They need to be trained and specialized.
Right. For T lymphocytes, this crucial preprocessing happens in the thymus gland.
The thymus.
Lymphoid progenitor cells destined to become T cells migrate from the bone marrow to the thymus.
Once in the thymus, they undergo rapid division and, crucially, develop immense diversity in their ability to react against different specific antigens.
The source notes this process generates thousands of distinct types of T lymphocytes, each with specific reactivity against a particular antigen. It's like mass-producing highly specialized keys, each designed to fit one unique lock the antigen.
Thousands of types. Incredible diversity from the start.
After this differentiation and diversification in the thymus, these preprocessed T lymphocytes are released and spread via the blood to populate lymphoid tissues throughout the body, ready for action.
And here is where one of the most vital functions of the thymus occurs, particularly important for preventing autoimmune disease. It ensures that the T lymphocytes it releases will not react against the body's own tissues.
Self-tolerance. This is huge.
How does it do this? The source describes a mechanism where developing T lymphocytes in the thymus are mixed with essentially all the body's own self-antigens.
Like a test.
Exactly. If a T lymphocyte shows any significant reactivity against these self-antigens, it's specifically targeted and destroyed by thymic epithelial cells and phagocytized. It fails the test.
And the source mentions that potentially up to 90% of the T cells generated in the thymus might be eliminated through this process.
90%. That's a massive quality control step, ensuring that only T lymphocytes that react only against foreign, outside antigens are allowed to leave the thymus. It teaches them not to attack self.
A brutal but necessary education. This self-tolerance education in the thymus primarily takes place shortly before and for a few months after birth.
So early life is critical for this.
Very critical. Removing the thymus later in life reduces T-cell immunity, but removing it before birth completely prevents the development of cell-mediated immunity.
So the thymus is the boot camp for T cells, training them for diversity and, crucially, teaching them who not to fight.
Well put. Now, for B lymphocytes, the pre-processing site in humans is the liver during mid-fetal development and the bone marrow during late fetal life and after birth.
Liver and bone marrow for B cells.
The source gives a historical nod to the bursa of Fabricius in birds, which is where B cells originally got their name, B for bursa.
Ah, okay. Key differences from T cells here.
Big ones. B lymphocytes don't become the direct attackers. They secrete antibodies, large protein molecules that circulate and combine with or destroy antigens. They send out the weapons.
And the source notes that B lymphocytes develop even greater diversity than T cells, capable of forming millions of distinct types of antibodies with different specific reactivities.
Millions. Even more diverse than T cells. After pre-processing, B lymphocytes also migrate to lymphoid tissue, lodging in areas adjacent to the T cell zones, ready to cooperate.
This distinction between B cells producing soluble weapons and T cells being the weapons themselves, along with their different processing sites, sets the stage for their distinct roles in acquired immunity.
Definitely. So how do these lymphocytes, pre-processed and ready, actually get activated by an antigen?
Yeah, the trigger mechanism.
The source explains that when a specific antigen enters the body and encounters its corresponding T or B lymphocyte clone in lymphoid tissue, that lymphocyte becomes activated.
And this activation leads to the rapid reproduction of that specific lymphocyte. The source introduces the crucial concept of lymphocyte clones here.
Right. Lymphoid tissue stores millions of different types of preformed, naive B and T lymphocytes, each representing a clone, a unique line of cells.
Each clone is capable of forming only one specific type of antibody or T cell with a single defined specificity.
Super specific. And only that single specific type of antigen can activate that clone. It needs the perfect match.
Once activated by its antigen, the lymphocyte reproduces rapidly, forming tremendous numbers of duplicates. This is clonal expansion.
Building an army quickly. Activated B lymphocytes will eventually differentiate and secrete antibodies, while activated T lymphocytes will become sensitized T cells.
A clone of lymphocytes, as defined by the source, includes all the different lymphocytes capable of forming one specific antibody or T cell. They are essentially identical, derived from a few early precursor cells of that type.
This clonal expansion is how the body amplifies a specific rare cell type into a massive force capable of dealing with a significant invasion, turning one soldier into a legion.
This brings up a fascinating puzzle the source addresses. How can a human genome, with only a few hundred to a few thousand genes dedicated to this process, possibly code for the millions of different types of antibodies and T lymphocytes that the body can create?
Yeah, it seems impossible if each specific antibody or T cell receptor required its own dedicated gene. You'd run out of genes fast.
So what's the trick?
The solution lies in gene segments. The source explains that the complete functional gene for forming a specific T cell receptor or antibody molecule is not preassembled in the original stem cells. Instead, there are hundreds of smaller gene segments, like Lego bricks.
Gene segments, okay.
During the preprocessing phase, in the thymus for T cells and bone marrow for B cells, these gene segments are mixed and recombined in random combinations within individual developing lymphocytes to form a complete functional gene.
So they shuffle the deck for each cell.
Pretty much. Because there are hundreds of types of gene segments and millions of different ways they can be shuffled and combined in individual cells, this random recombination process can generate millions of different unique gene structures. Each mature functional T or B lymphocyte ends up with a gene structure that codes for only a single antigen specificity. These highly diverse, pre-coded lymphocytes then populate the lymphoid tissues, ready to encounter their specific antigen.
It's like having an alphabet of gene segments, and each lymphocyte creates a unique word or sentence by randomly picking and arranging these segments. That single unique word is its specific recognition code for one antigen.
That's a great analogy. This elegant mechanism of random gene segment recombination followed by selection is key to the incredible diversity and specificity of the acquired immune system. It's genetic origami.
So we have these millions of diverse clones waiting. How does an antigen actually activate its specific clone? The source tells us each clone is responsive to only a single type of antigen because of specific receptors.
Right. B lymphocytes have a remarkable number of antibody molecules embedded in their cell surface membrane, about 100,000 per cell.
100,000 receptors on one cell.
Yeah. These surface antibodies function as receptors. They are highly specific and will react with only one type of antigen. When that antigen binds to these surface antibodies, it's the primary trigger for the B cell's activation process.
And T lymphocytes have similar molecules on their surface called surface receptor proteins, or T cell receptors.
Also highly specific, like the B cell receptors. A single T cell can have as many as 100,000 of these receptor sites too.
So the presence of complementary receptors that can bind to a specific antigen is what ensures that only the correct specific lymphocyte clone is stimulated to respond, lock and key again.
Exactly. But the source adds another layer to this activation story, highlighting the vital role of macrophages.
The big eaters.
Right. Macrophages are everywhere in lymphoid tissue, strategically positioned near the lymphocytes. When invading organisms enter, they are often first engulfed and partially digested by these macrophages through phagocytosis.
They clean up, but they do more.
Oh, yes. The macrophages then process these invaders and liberate the antigenic products into their internal environment. Crucially, the macrophages then present these antigens directly to the lymphocytes through cell-to-cell contact.
Antigen presentation.
This act of presenting the processed antigen is essential for activating the specific clones of T and B lymphocytes. They basically show the lymphocytes what the enemy looks like.
And it's not just passive presentation, right?
No, macrophages also secrete a special activating substance called interleukin-1. This interleukin-1 acts as a signal that further promotes the growth and reproduction of the specific lymphocytes that have been presented with the antigen.
So they present the evidence and provide a boost.
Exactly. Macrophages are not just cleaners, they are key presenters and activators in this whole process.
And there's another critical helper in activating B cells, T cells themselves. The source notes that most antigens activate both T and B lymphocytes simultaneously.
Right. It's often a coordinated response. Some of the T cells that are activated and proliferate are called T helper cells.
T helper cells. We'll talk more about them soon.
These T helper cells secrete protein mediators called lymphokines, signal molecules.
And these lymphokines are essential co-stimulators for B cells.
Very essential. Without the aid of lymphokines secreted by T helper cells, the quantity of antibodies formed by B lymphocytes in response to an antigen is usually quite small. They need that help signal.
This shows a crucial cooperative relationship. T helper cells are often required to provide the necessary signals, lymphokines, to ensure a robust antibody-producing response from B lymphocytes. Teamwork makes the dream work.
It really does in the immune system.
Let's take that detailed understanding of B cell activation and dive specifically into humoral immunity, the B lymphocyte system, and the powerful antibodies it produces. How do we get from a stimulated B cell to billions of antibody molecules?
Okay, yeah, the production line. The source walks through the formation of antibodies by plasma cells. Before antigen exposure, specific B-cell clones exist as dormant, naive cells in lymphoid tissue, just waiting.
When a foreign antigen enters, macrophages phagocytize and present it to nearby B and T cells.
Yeah.
This presentation, combined with help from activated T helper cells and their lymphokines,
That essential help signal.
leads to the intense activation of the specific B lymphocytes. They wake up.
These activated B lymphocytes then enlarge and transform into rapidly dividing cells called lymphoblasts.
Lymphoblasts, okay.
Some of these lymphoblasts differentiate into plasmablasts, which are the precursors to antibody-producing plasma cells. These plasmablasts undergo dramatic cellular changes.
Like what?
Notably, a vast proliferation of their rough endoplasmic reticulum, the cellular machinery responsible for synthesizing large protein molecules like antibodies. They gear up for mass production.
These plasmablasts then divide incredibly rapidly, roughly every 10 hours for about 9 divisions.
That's fast. So from a single plasmablast, you can get a population of about 500 cells within just 4 days. Exponential growth.
And the mature plasma cells are phenomenal antibody factories. The source states that each plasma cell can produce gamma globulin antibodies at an extremely rapid rate, about 2,000 molecules per second.
2,000 per second per cell. That is just staggering output.
These antibodies are then secreted into the surrounding lymph, collected, and eventually enter the circulating blood,
Flooding the system with protection. This intense antibody production continues for several days or weeks until the plasma cells essentially exhaust their capabilities and undergo programmed cell death. They burn out.
But not all activated lymphoblasts become short-lived plasma cell factories. The source emphasizes the formation of memory cells, which are crucial for enhancing future responses.
Ah, yes, the memory B cells. A few of the lymphoblasts generated during the initial activation of a B cell clone don't differentiate into plasma cells. Instead, they become new B lymphocytes, similar to the original ones, but now designated as memory cells.
This process significantly increases the total population of that specific B cell clone. These memory cells circulate throughout the body and populate all the lymphoid tissues.
They remain immunologically dormant, waiting, until they encounter the same antigen again.
And the payoff is huge. When that subsequent exposure happens, these numerous memory cells are quickly activated, leading to a much more rapid and far more potent antibody response compared to the first time the body saw the antigen.
Faster and stronger the second time around. The source illustrates this with the primary versus secondary antibody response curve.
Right, I remember seeing figures like that. The primary response on first exposure typically has a lag phase of about a week before detectable antibodies appear, is relatively weak in the amount of antibody produced, and the antibody level declines quickly.
But the secondary response, after re-exposure, starts much faster, sometimes within hours. It's far more potent, reaching significantly higher antibody concentrations, and the antibody production continues for many months, leading to longer-lasting protection.
This powerful secondary response is the fundamental principle behind vaccination schedules. Giving multiple doses of a vaccine over time
The booster shots
leverages the memory cell system to build a robust, long-lasting protective antibody level.
Precisely. And the source clarifies that the plasma cells themselves contribute to both the immediate response and a long-term immunity. Activated B cells differentiate into short-lived or long-lived plasma cells.
So 2 types of plasma cells.
Seems so. The short-lived ones are the rapid responders, pumping out antibodies intensely for days before dying. But the long-lived plasma cells reside in places like the bone marrow and gut-associated lymphoid tissue and continue producing antibodies for many years.
For years. Wow. This population of long-lived plasma cells is what provides that lifelong immunity we see for diseases like measles or smallpox.
Yeah, the source gives remarkable examples. Detectable levels of smallpox antibodies 70 years after vaccination, and functional 1918 H1N1 antibodies found in survivors 90 years later.
90 years later. That persistence of antibody production from long-lived plasma cells is truly incredible. Immune memory is long.
It really is. So we understand how they're made, but what exactly are these antibodies structurally?
Yeah, the molecules themselves.
The source describes their nature as gamma globulins, also known as immunoglobulins or IGs. They are large protein molecules making up about 20% of plasma proteins, with molecular weights ranging from 160,000 to 970,000. Pretty hefty.
They're all composed of light and heavy polypeptide chains. The most common structure, like IgG, consists of 2 light chains and 2 heavy chains arranged in a Y shape.
The classic Y structure. However, some immunoglobulins are much larger, containing up to 10 light and 10 heavy chains, forming more complex structures like pentamers, but they always have at least 2 and up to 10 heavy light chain pairs.
Within this structure, there's a critical distinction between the variable and constant portions of the polypeptide chains.
Very important distinction. The variable portions are located at the tips of the Y-arms and are unique to each specific antibody molecule. This is the part that attaches specifically to a particular antigen, the business end.
And the constant portions form the rest of the molecule, including the stem of the Y. These constant portions determine other biological properties of the antibody,
like what class it is, how it interacts with other cells or complement,
such as how it diffuses in tissues, its ability to attach to cells or the complement system, how it's transported across membranes, and what class of antibody it is. It dictates the function beyond just binding.
Right. The light and heavy chains are linked together by both non-covalent and covalent disulfide bonds, holding the structure together.
The source goes into detail about the specificity mechanism, how antibodies bind to antigens with such precision.
It's all about shape. This is due to the unique three-dimensional structure formed by the amino acid sequences in the variable portions of the light and heavy chains.
This unique structure creates a binding site that is complementary in shape to a specific epitope on the antigen. The prosthetic grooves on the antigen surface fit into the binding site of the antibody like a mirror image.
Or like a key in a lock. When the antibody is highly specific for an antigen, there are many points of contact and bonding sites, leading to an exceedingly strong coupling.
What kind of bonds?
The forces involved include hydrophobic interactions, hydrogen bonds, ionic attractions, and van der Waals forces. Lots of weak bonds adding up to strong specific binding.
The tightness of this binding is quantified by the affinity constant, Ka, which measures the strength of the antibody-antigen interaction based on the law of mass action. A higher Ka means tighter binding.
Right. The source also highlights the bivalent nature of antibodies like IgG, meaning they have 2 identical antigen binding sites.
2 arms on the Y.
Exactly. This allows them to bind to multiple antigen molecules or cross-link multiple particles, which is important for mechanisms like agglutination. Some larger antibodies like IgM can have up to 10 binding sites, making them very good at grabbing things.
The source lists the 5 major classes of antibodies, or immunoglobulins: IgM, IgG, IgA, IgD, and IgE, often remembered by the mnemonic GAMED.
GAMED, yeah. While they all serve immune functions, the source particularly highlights IgG, which is the most abundant class, about 75% of antibodies, and a major player in defense.
The workhorse.
And IgE, which is present in small amounts but is critically important in allergic reactions, sometimes called regions or sensitizing antibodies. We'll definitely come back to IgE.
IgM is also noted as being a significant component of the primary immune response.
The first responder antibody type.
And is often very effective due to its structure, which can have up to 10 binding sites, allowing it to bind strongly and cross-link antigens, even when its overall concentration might be lower than IgG in later responses. It's like throwing out a big net.
Good analogy. So these are the structures and types of antibodies produced by B cells. But how do they actually do their job of protecting the body?
Right, the mechanisms of action. The source outlines 2 main ways antibodies work.
First, there's direct attack on the invader. Second, and usually more powerful, is the activation of the complement system, which then launches a multi-pronged assault.
Let's look at the direct actions first. Because antibodies are often bivalent and antigens frequently have multiple epitopes, antibodies can bind to invaders in ways that lead to their inactivation or clearance.
Right. This includes agglutination, where antibodies bind to and clump together multiple large particles like bacteria or red blood cells,
making them easier targets for phagocytic cells, right?
Yeah.
Easier to swallow a clump.
Exactly. Precipitation is another direct mechanism where soluble antigens, like toxins, bind to antibodies, forming large insoluble complexes that precipitate out of solution and can be cleared, takes them out of circulation.
Antibodies can also directly neutralize toxins or viruses by binding to their active or binding sites, covering them up, and rendering the invader non-virulent or non-toxic, basically blocking their function.
Simple but effective, and in some cases, particularly potent antibodies can directly attack and rupture the membranes of cellular invaders through lysis. However, the source notes that these direct actions alone are often insufficient to provide major protection. They need backup.
That's where the complement system comes in, acting as a massive amplifier of antibody action.
The cavalry arriving. The complement system, as we briefly mentioned under innate immunity, is a complex cascade of about 20 proteins, many of which are enzyme precursors, present in blood plasma and tissue fluid in an inactive state, just waiting.
Its main function is to complement or enhance the actions of antibodies and phagocytes. It helps neutralize and destroy pathogens, clear damaged cells, and promote inflammatory responses.
The source details the classical pathway of complement activation, which is initiated by an antigen-antibody reaction. When an antibody binds to an antigen, it causes a conformational change that exposes or activates a specific site on the antibody's constant portion.
The stem of the Y changes shape.
Essentially, yes. This activated constant region then binds directly to the first complement molecule, C1. This binding initiates a chain reaction, a cascade of enzyme activation, dominoes falling.
The source emphasizes that this cascade is designed for amplification. An activated enzyme in an early step activates a larger quantity of enzymes in the next step, and so on,
Leading to a huge, amplified reaction starting from a relatively small initial antigen-antibody binding event. A little spark starts a big fire.
This cascade generates numerous end products, which are the molecules that carry out the crucial functions of the complement system in preventing tissue damage and clearing invaders.
Let's break down some of the most important effects of the complement cascade listed in the source. First, opsonization and phagocytosis.
Opsonization, making things tasty for phagocytes.
Pretty much. A key complement fragment, C3b, is a potent opsonin. It binds to bacteria and other particles that have antibody-antigen complexes attached.
So it coats the tagged invader.
Right. C3b strongly activates neutrophils and macrophages, causing them to avidly engulf and digest the tagged bacteria. The source states this can increase the rate of bacterial destruction by hundreds of times. It's like applying a chemical marker that screams, "Eat me," to the phagocytes.
Huge amplification. What else?
Another crucial effect is lysis, direct killing. One of the most important end products is the membrane attack complex, MEC.
MEC.
Yeah. Formed by a combination of complement factors, C5b, C6, C7, C8, and multiple C9 molecules, C5b, 6, 7, 8, 9.
This MAC complex inserts itself into the lipid bilayer of the cell membrane of bacteria or other target cells. This creates pores.
Essentially punching holes in the membrane, big holes.
These pores make the membrane permeable to ions and water. Fluid rapidly flows into the cell from the surrounding interstitial space, causing the cell to swell greatly and usually rupture shortly after due to osmotic lysis, POP.
A very effective direct killing mechanism. Complement products also contribute to agglutination, modifying the surfaces of invaders, causing them to stick together and clump, aiding clearance.
They can neutralize viruses by binding to and damaging their structures, rendering them non-virulent.
Chemotaxis is another vital function. The complement fragment C5a is a powerful chemoattractant. It calls for backup.
How?
It initiates the migration of large numbers of neutrophils and macrophages from the blood into the tissue area where the antigenic agent is located, directing the cellular cleanup and attack force like a chemical flare.
Complement fragments, C3a, C4a, and C5a, also act as anaphylatoxins. They activate mast cells and basophils,
The allergy cells. We'll see them again.
causing them to degranulate and release substances like histamine, heparin, and other mediators into the local tissues.
These mediators cause increased local blood flow, increased capillary permeability leading to fluid and protein leakage into tissues, and other local reactions that help contain and inactivate the antigen. Inflammation, basically.
The source notes these factors are significant players in both inflammation and allergy.
And finally, other complement products enhance inflammatory effects by further increasing blood flow and capillary permeability and promoting the coagulation of interstitial fluid proteins. This coagulation can help physically wall off the area, preventing the spread of the invading organism through the tissues containing the threat.
So the complement system isn't just one mechanism. It's a complex, amplifying cascade that translates a single antibody-antigen binding event into a series of powerful defensive actions. Tagging for destruction, direct killing by membrane puncture, clumping, neutralization, cell recruitment, and local containment.
It's a true force multiplier for the humoral response. Antibodies point the way. Complement delivers a lot of the heavy ordnance.
Now let's turn to the other main arm of acquired immunity, the cell-mediated system, driven by T lymphocytes and their activated T cells.
Right, the cellular fighters. Just as B lymphocytes proliferate and differentiate into plasma cells and memory cells after antigen exposure, T lymphocytes also undergo clonal expansion upon encountering their specific antigen.
which is typically presented by macrophages, as we said.
Yes. Large numbers of these activated, specifically reacting T cells are then released from the lymphoid tissue. Unlike antibodies, which are secreted molecules, here the whole activated cells are released.
The cells themselves are the weapon.
They enter the lymph, flow into the blood, circulate throughout the body, can exit capillaries into tissue spaces, and continue circulating, some persisting for months or even years. Patrolling.
And like B cells, T lymphocytes also form memory cells. A portion of the activated T cell clone is preserved as new T lymphocytes that populate the lymphoid tissue throughout the body.
These T memory cells provide a similar benefit to B memory cells. Subsequent exposure to the same antigen anywhere in the body triggers a far more rapid and powerful release of activated T cells compared to the primary response.
Faster, stronger cellular response the second time.
Exactly. It's the cellular arm's way of remembering the enemy and responding faster and stronger next time.
The source emphasizes that T cell responses are extremely antigen-specific and are vital for defense. In fact, most acquired immune responses, even antibody production, usually require T cell assistance to initiate and become robust.
They're crucial helpers, not just killers. But here's a crucial difference in how T cells recognize antigens compared to B cells.
What's that?
T lymphocytes generally respond to antigens only when those antigens are bound to specific molecules called MHC proteins on the surface of antigen-presenting cells, APCs, within the lymphoid tissues.
MHC proteins. They don't interact directly with free-floating antigens like B cells do?
Generally, no. They need that formal presentation on an MHC molecule. The source lists the three major types of APCs: macrophages, which we already saw, B lymphocytes. Interesting that they can also present antigens.
B cells can present to T cells.
Yep, part of that cooperation. And dendritic cells.
Dendritic cells are highlighted as the most potent APCs. They're found throughout the body, and their primary function is specifically presenting antigens to T cells. They're professional presenters.
For a T cell to get activated, it needs to bind to the APC displaying the antigen-MHC complex for a sufficient duration. This stable interaction is facilitated by cell adhesion proteins on the surfaces of both cells. They need to connect properly.
The MHC proteins themselves are encoded by a large gene complex called the Major Histocompatibility Complex, MHC. Their function is to bind peptide fragments derived from proteins that have been degraded inside the APC.
So they chop up the invader protein?
And display the pieces. These fragments are then transported to the APC surface and displayed within the groove of the MHC molecule for T cells to recognize, like putting a sample on a display stand.
There are two main classes of MHC proteins involved in antigen presentation to T cells. MHC class I proteins present antigens to cytotoxic T cells.
The killer T cells.
While MHC class II proteins present antigens to T helper cells. This ensures the right type of T cell sees the antigen and gets the right message.
T lymphocytes have their specific antigen receptor molecules on their surface, similar to antibodies. These receptors have a variable unit that binds specifically to the antigen fragment displayed by the MHC protein.
That's the specific recognition part. This variable unit provides the specificity, while the rest of the receptor molecule is anchored to the T cell membrane. As with B cells, a single T cell can have up to 100,000 of these receptors.
So the T cell acts like a security officer who only recognizes the suspect when shown their ID photo, antigen fragment, held up by an authorized officer, MHC protein, from the police force, APC.
That's a good way to visualize it. And just as there are different types of B cell clones making different antibodies, there are different types of T cells, classified into three major functional groups with distinct roles in cell-mediated immunity.
These are T helper cells, cytotoxic T cells, also called killer cells, and regulatory T cells, also called suppressor T cells.
Let's start with T helper cells. They're typically the most numerous, making up over 75% of all T cells. As their name implies, they're crucial helpers.
How do they help?
The source describes them as the major regulator of virtually all immune functions. They perform this regulatory role by producing and secreting protein mediators called lymphokines.
Lymphokines, again, the signaling molecules.
These lymphokines act as signaling molecules that stimulate the growth, proliferation, and differentiation of other cells of the immune system, including cytotoxic T cells, B cells, and macrophages, as well as bone marrow cells that produce immune cells. They're like the conductors of the orchestra.
The critical importance of T helper cells is underscored by what happens in their absence. The source states that without the lymphokines from T helper cells, the rest of the acquired immune system is almost entirely paralyzed.
Paralyzed, that's strong.
This is tragically demonstrated by the effects of HIV infection. The human immunodeficiency virus primarily targets and destroys T helper cells.
Right, that's its main target.
This loss leaves the body unable to mount effective acquired immune responses, making individuals susceptible to opportunistic infections and cancers that a healthy immune system would easily handle, leading to AIDS. It truly shows they are central command.
Absolutely devastating and highlights their central role. The source briefly mentions that naive CD4 plus T helper cells can differentiate into distinct subsets upon stimulation, such as Th1, Th2, and Th17 cells.
Different types of helpers.
Yeah, specializing further. These subsets produce different panels of lymphokines and steer the immune response towards different types of reactions, like activating macrophages for clearing intracellular pathogens, Th1, stimulating antibody production, especially IgG and IgE, Th2, or recruiting neutrophils and monocytes to fight extracellular bacteria and fungi, Th17, tailoring the help provided.
Looking specifically at the regulatory functions of lymphokines, the source details several. First, they stimulate the growth and proliferation of cytotoxic T cells and regulatory T cells.
Right. Without T helper lymphokines, activation of these cells by antigen is only slight. Interleukin-2, a key lymphokine from T helper cells, is noted as being an especially strong stimulator of their growth. IL-2 is like fuel for T cells.
Second, lymphokines potently stimulate B cell growth and differentiation into plasma cells and antibodies. Direct antigen action on B cells is weak without T helper cells.
We saw this earlier. Many interleukins participate, particularly IL-4, IL-5, and IL-6, which are so important for B cells they're sometimes called B cell stimulating or growth factors. This reinforces the crucial T helper cell requirement for a strong humoral response.
Third, lymphokines activate the macrophage system.
How?
They cause macrophages to accumulate in inflamed tissue by slowing their migration. More importantly, they activate macrophages, making them much more efficient at phagocytosis and destruction of invading organisms, supercharging the cleaners.
And finally, some lymphokines, especially interleukin-2, provide a positive feedback loop, stimulating T helper cell activation itself.
Boosting themselves.
Yeah. This serves as an amplifier, boosting the helper cell population and signal output, thereby enhancing the entire immune response, ramping things up.
So T helper cells are truly the central coordinating hub and amplifier of the acquired immune system, using their lymphokine signals to direct and boost the activities of cytotoxic T cells, B cells, and macrophages. Absolutely essential.
Now, let's move to the cytotoxic T cells, the killer cells. These are the direct executioners of the cell-mediated system.
The hitmen.
They are capable of killing targeted cells, including infected body cells, cancer cells, transplanted cells, and sometimes microorganisms directly.
How do they do it? CD8 plus cytotoxic T cells have specific receptor proteins that allow them to bind tightly to the target cell or organism, displaying the appropriate antigen presented on MHC class I proteins.
They lock onto the target. Once bound, the cytotoxic T cell acts rapidly. It secretes hole-forming proteins called perforins.
Perforins, they perforate.
Exactly. These perforins insert into the membrane of the target cell, literally punching molecular holes in it.
This disruption of the membrane allows fluid from the surrounding interstitial space to rapidly flow into the target cell, causing it to swell dramatically.
Like popping a water balloon. In addition to perforins, the killer cell also injects or releases other cytotoxic substances directly into the attacked cell, such as granzymes, which can trigger programmed cell death, apoptosis.
So holes in toxic substances.
A deadly combination. The combined effect is that the attacked cell usually swells and dissolves shortly after. Mission accomplished.
A remarkable detail is that these killer cells are efficient and mobile. After initiating the killing process on one target cell, they can detach and move on to bind and kill other cells carrying the same antigen.
Serial killers, in a good way. Some can persist for months in the body, providing ongoing surveillance.
They are particularly effective against cells infected by viruses because viral antigens are often displayed on the surface of the infected cell, making them targets for cytotoxic T cells.
Right, they spot the infected cells. They are also the primary mechanism for recognizing and destroying cancer cells that display abnormal surface antigens and for rejecting transplanted organs or tissues that are recognized as foreign. Critical roles.
The third group are regulatory T cells, sometimes called suppressor T cells. The source indicates that our understanding of these cells is less extensive than for helper and cytotoxic T cells.
Yeah, Treg cells are still being actively researched. However, their function is quite important. They are capable of suppressing the activity of both cytotoxic T cells and T helper cells.
Putting the brakes on. Why?
The proposed purpose of this suppression is to prevent excessive immune reactions that could damage the body's own tissues, keeping the response proportionate and preventing friendly fire.
It's believed that regulatory T cells play a significant role in maintaining immune tolerance, limiting the immune system's ability to attack self-tissues. This brings us to a critical concept.
Immune tolerance. It's the flip side of powerful specificity. If acquired immunity is so powerful and specific, it could potentially be devastating if it turned against the individual's own body tissues.
Yeah, that seems like a major risk.
Fortunately, the immune system normally recognizes the body's own tissues as distinct from foreign invaders. It typically forms very few antibodies or activated T cells against self-antigens.
The source explains that this critical tolerance primarily develops during the pre-processing phase of lymphocytes in the thymus, for T cells, and bone marrow, for B cells. Back to that early training.
Right. Experimental evidence supports this. Injecting a strong antigen into a fetus during the period when lymphocytes are being pre-processed in these organs can actually prevent the development of specific lymphocyte clones for that antigen in the adult.
So you can induce tolerance early.
The immune system is essentially taught tolerance to that antigen early on. The proposed mechanism, especially for T cells in the thymus, involves exposing immature lymphocytes that are potentially reactive against self-antigens to those self-antigens during pre-processing.
The self-antigen test again.
These self-reactive lymphocytes become activated, proliferate, interact with the self-antigen, and are then believed to be destroyed by specialized epithelial cells in the thymus before they can mature and migrate to other lymphoid tissues. Negative selection.
The conclusion is that this negative selection process during pre-processing in the thymus and bone marrow eliminates most, if not all, clones of lymphocytes that would otherwise attack the body's own tissues. They are self-destroyed due to continual exposure to self-antigens in this critical developmental window.
A crucial safety mechanism. But sometimes this vital tolerance mechanism fails. When a person loses immune tolerance to their own tissues, autoimmune diseases can occur.
Autoimmunity, when the body attacks itself.
The source notes that this failure seems to happen more frequently as people age. It's often triggered after some destruction of the body's own tissues, which releases quantities of self-antigens into the circulation.
Like after an injury or infection.
Potentially. This sudden influx of self-antigens presumably bypasses or overwhelms the tolerance mechanisms, triggering an acquired immune response, either activated T cells or antibodies against self.
Over 100 different autoimmune diseases have been identified. The source provides several specific and impactful examples.
Rheumatic fever is one. This occurs after infection with a specific type of Streptococcus bacteria. The bacterial toxin has an epitope that is molecularly very similar to certain self-antigens found in the heart and joints, particularly heart valves.
Molecular mimicry. The immune system gets confused.
Exactly. The immune response aimed at the strep toxin mistakenly targets these self-tissues as well, causing damage. It's a classic case of molecular mimicry causing self-attack.
One type of glomerulonephritis is another example, caused by the immune system becoming reactive against the basement membranes of the kidney glomeruli, impairing kidney function.
Myasthenia gravis involves immunity against the acetylcholine receptor proteins at the neuromuscular junction. Antibodies bind to and block these receptors, disrupting nerve signals to muscles and causing profound weakness and paralysis.
Multiple sclerosis, MS, is described as a disease where the immune system attacks the myelin sheath, the insulating layer covering nerve fibers in the brain and spinal cord.
Demyelination. This disrupts nerve signal transmission, leading to a wide range of neurological symptoms.
And systemic lupus erythematosus, SLE, is a severe, widespread autoimmune disease where the immune system generates antibodies against antigens found in many different body tissues simultaneously,
attacking multiple systems at once,
leading to extensive inflammation and damage that can be fatal in severe cases. These examples really bring home the devastating consequences when the system designed to protect misidentifies self as enemy.
It's a powerful system, and when it goes wrong, it can be really destructive. Given the incredible power and specificity of acquired immunity, it's a natural step to intentionally harness it for protection.
Right. This is the principle behind immunization, achieved by injecting antigens.
Immunization has been a cornerstone of public health for centuries, aiming to produce acquired immunity against specific infectious diseases or toxins without causing the full, potentially dangerous disease itself, training the immune system safely.
The method involves injecting antigens. The source describes types of active immunity induced by immunization, where the person's own immune system is stimulated to produce antibodies or activated T cells.
One method uses dead organisms. These organisms are killed so they cannot cause the disease, but they still retain their chemical antigens.
Like killed vaccines. Examples include vaccines for typhoid fever, whooping cough, and diphtheria, using the toxoid.
Another method uses treated toxins called toxoids. The toxic component of a bacterial toxin is chemically inactivated, but the antigenic structure remains intact.
So you become immune to the toxin, not the bacteria itself.
Right. Injecting toxoids induces immunity to the toxin. Tetanus and botulism vaccines use toxoids.
And a powerful method uses attenuated live organisms. These are live organisms that have been weakened, attenuated,
Skin but still alive.
through methods like growing them in special culture media or passing them through animals repeatedly. They retain the specific antigens needed to induce immunity, but have mutated into non-disease-causing forms.
Examples include vaccines for smallpox, yellow fever, poliomyelitis oral vaccine, and measles. These often give very strong, long-lasting immunity.
In all these active immunization methods, the body's own immune system is doing the work, generating memory cells and long-lived plasma cells for sustained protection, building its own defenses.
There's also a way to get immediate but temporary protection through passive immunity. This involves achieving immunity without injecting antigen into the recipient.
How does that work? Borrowing immunity?
Essentially, yes. Temporary immunity is conferred by infusing preformed antibodies, activated T cells, or both, obtained from the blood of another individual, human or animal, who has already developed active immunity against the specific antigen.
Like getting an infusion of antibodies.
Exactly. The duration of this passive protection is limited, though. Antibodies typically persist in the recipient's circulation for about 2 to 3 weeks, providing protection during that time.
And T cells.
Activated T cells transferred from another human might last a few weeks, but those from an animal are often cleared rapidly, lasting only hours or days. The body recognizes them as foreign, too.
The key benefit of passive immunity is speed. It provides immediate protection, which can be life-saving in situations where the individual has been exposed to a deadly toxin or pathogen, and there isn't time for their own immune system to develop active immunity through vaccination.
A critical emergency measure sometimes. So we've covered the incredible protective capabilities of the immune system, both innate and acquired. But as the source notes, sometimes immunity has undesirable side effects, leading to conditions like allergy or hypersensitivity reactions.
The downside. The source highlights that some types of allergy, particularly atopic allergies, occur in people with a specific genetic predisposition, what's called a non-ordinary response of the immune system.
A tendency towards allergy. There are several types of these reactions. One important type is delayed reaction allergy. The source makes it clear this type is caused by activated T cells, not antibodies.
Cell-mediated allergy. The classic example is poison ivy. The toxin from the plant isn't inherently destructive, but in sensitive individuals, repeated exposure triggers the formation of activated helper and cytotoxic T cells that are specific for antigens related to the toxin.
So the first exposure sensitizes the T cells.
Right. Upon subsequent contact with poison ivy, these pre-sensitized T cells migrate to the skin area. After a delay of about a day or so, they initiate a cell-mediated immune reaction in the tissue.
Hence, delayed reaction. As we discussed with cell-mediated immunity, this involves the release of toxic substances from the activated T cells and the recruitment and activation of macrophages.
The result, in the case of poison ivy, is serious tissue damage in the skin, the characteristic rash, blistering, and inflammation. It's the immune response causing the damage, not the plant toxin directly.
A really important point. This same mechanism can occur in response to other antigens, like those inhaled. In the lungs, it can lead to tissue damage, edema, and potentially asthmatic attacks.
The other major type of allergy discussed is atopic allergies, strongly associated with the presence of excess IgE antibodies. These occur in genetically predisposed individuals.
IgE, the allergy antibody. The key feature is unusually high levels of IgE antibodies in the blood. These IgE antibodies are also called regions or sensitizing antibodies because they have a very strong tendency to attach to the surface of mast cells and basophils.
They coat these cells how many?
The source states that a single mast cell or basophil can bind up to 500,000 IgE molecules. They become heavily coated with these potential reaction triggers, like little landmines.
Wow. Then when an allergen, which is an antigen that reacts specifically with the IgE antibodies coating the mast cells, enters the body, it binds to multiple IgE molecules on the surface of a sensitized mast cell or basophil.
Cross-linking the IgE receptors. This allergen region binding causes an immediate change in the mast cell or basophil membrane, sometimes causing the cells to rupture, but more often triggering them to rapidly release powerful chemical mediators that were stored inside them. Degranulation.
What kind of mediator?
A whole cocktail. Histamine, protease, the slow-reacting substance of anaphylaxis, which is a complex mixture of toxic leukotrienes, eosinophil and neutrophil chemotactic substances, heparin, and platelet-activating factors.
A chemical storm. These substances then act on surrounding tissues, causing a variety of effects. Dilation of local blood vessels, increased permeability of capillaries leading to fluid leakage into tissues, attraction of other immune cells like eosinophils and neutrophils, and contraction of local smooth muscle cells.
The classic allergy symptoms. The specific symptoms of the allergic reaction depend heavily on where the allergen region reaction occurs. The source gives several examples of specific atopic reactions.
Anaphylaxis is the most severe and potentially life-threatening. It's a widespread allergic reaction that happens when an allergen enters the circulation rapidly, perhaps by injection or rapid absorption from the gut.
Systemic reaction. This triggers a massive release of mediators from basophils in the blood and mast cells throughout tissues that are coated with IgE regions.
What happens then?
The histamine released causes widespread vasodilation and dramatically increased capillary permeability throughout the body. This leads to a massive loss of plasma volume from the blood into the tissues.
Which causes shock.
Exactly. The source warns that this can cause the person to die of circulatory shock within minutes if not treated, typically with epinephrine, which constricts vessels and relaxes airways. It's a medical emergency.
Simultaneously, the slow-reacting substance of anaphylaxis, the leukotrienes, is released. These cause severe spasm of the smooth muscle in the bronchioles of the lungs,
Making it hard to breathe.
leading to a severe asthma-like attack and potentially death by suffocation. Anaphylaxis is a terrifying display of a systemic, uncontrolled immune response.
Truly scary. Urticaria, or hives, are localized anaphylactoid reactions that occur when antigen enters specific areas of the skin.
Like a bug bite reaction.
Could be. Local histamine release causes immediate vasodilation, producing a red flare, and increased local capillary permeability, leading to local circumscribed areas of swelling within minutes, the hives or wheals.
The source notes that antihistamine drugs can prevent hives by blocking histamine's effects, indicating histamine is a primary mediator in this localized reaction.
Hay fever is another common example, where the allergen region reaction happens in the nose, often triggered by pollen or dust mites.
Seasonal allergy.
Right. Histamine release causes local vascular dilation in the nasal passages, increasing capillary pressure and permeability. This rapid fluid leakage into the nasal cavities and tissues leads to the characteristic swollen, congested, and excessively secretory nasal linings. Runny nose, itchy eyes.
The source mentions antihistamines can help reduce the swelling, but other mediators still cause irritation and sneezing. They don't block everything.
Finally, asthma frequently occurs in hypersensitive allergic individuals. In asthma, the allergen region reaction primarily takes place in the bronchioles of the lungs.
So deeper in the airways.
Yes. The slow-reacting substance of anaphylaxis, leukotrienes, is believed to be a particularly important mediator released here, causing severe spasm of the smooth muscle in the walls of the bronchioles.
This constriction makes breathing extremely difficult. Wheezing, shortness of breath.
The source points out that antihistamine medication is less effective in treating asthma compared to hives or hay fever because histamine is not the major factor driving the smooth muscle spasm. Leukotrienes play a more significant role. Need different treatments like bronchodilators or anti-leukotrienes.
So allergies, particularly atopic allergies, represent a powerful demonstration of how the immune system's machinery, when directed by IgE antibodies and triggered in sensitive individuals, can unleash a storm of mediators that cause significant tissue dysfunction and even life-threatening reactions. The friendly fire aspect again.
Exactly. It's the system overreacting inappropriately.
Wow. We've journeyed through an incredible amount of detail here, peeling back the layers of this complex system. We've seen the immediate, broad protection of innate immunity.
And first line.
And contrasted it with the astonishingly powerful and specific acquired immunity.
The adaptive learning system. We've delved into how antigens trigger specific lymphocyte clones, the critical pre-processing steps in the thymus and bone marrow, and the distinct roles of T and B lymphocytes.
The cellular players. We've explored the intricate mechanisms of antibody action amplified by the complement cascade.
Antibodies and their backup.
And the direct cellular attack launched by activated T cells.
The cell-mediated killers. We've also confronted the vital process of immune tolerance, how the body learns not to attack itself, and what happens when that tolerance breaks down in the array of autoimmune diseases.
Protecting self versus attacking self. Crucial balance.
And we've seen how this system is leveraged for health through immunization and how it can sometimes go awry in allergic hypersensitivity.
The good, the bad, and the overreactive. Understanding these mechanisms at this level of detail really does feel like getting a grasp on what makes this system operate like the elite professionals study it.
It's appreciating the sheer complexity and efficiency required to defend a complex organism like the human body. It's truly amazing engineering.
Exactly. Considering the elegance of gene segment recombination to create millions of specific receptors, the vital cooperation between different cell types and molecules, the amplification cascades, and the cellular armies on patrol, it's truly mind-boggling biological engineering.
It makes you think about the sheer robustness and adaptability of life, doesn't it? And it leaves so much room for further discovery.
Always more to learn.
How can we better understand these intricate controls, enhance the desired responses, suppress the undesirable ones, and truly harness this power for human health in ways we haven't even imagined yet?
Absolutely. Something to ponder. Indeed, now that you've taken this deep dive into your own incredible internal defense system.